This is a short guide on experimental design for RNA sequencing experiments. So the suggested reading on the following slide, which is highly recommended. The first resource, Hitchhiker's Guide to Expression Analysis, goes into very deep detail on all aspects of RNA sequencing data and is very comprehensive and very good reading. A second article is recent work demonstrating why certain tools, known as HR and DE-seq2, that are commonly used, but are nonetheless give very high false positive rates, such that if you use a threshold of, say, p is equal to 0.05, in practice, this actually translates to a much higher threshold of p is equal to zero point 0.2, with the consequent effects of having far higher rates of false positives and much less positive predictive power as a consequence. So that's why these two tools are not recommended at all. There's a further guide here by Busby on RNA-seq experiments, and we'll later use this application, a web application, or an RNA-seq site size calculation. And there's further resources here on experimental design. And then it's suggested that you also consider looking at the omics learning courses ST2 on hypothesis testing and G06 on transcriptomics if you want to explore this topic further. What we're going to discuss here is firstly how biological technical replicates inform RNA sequencing experiments. And secondly, we're considering how we can model the power in different RNA sequencing experimental designs in different organisms. What we first need to think about when we're doing RNA sequencing is how we actually apply it and the numerous steps involved. So firstly, we have the nuances of this stage of experimental design, and then considering how we're going to collect samples in a manner that informs our experimental question. We have to isolate the mRNA, which again, RNA is very fragile. They degrade very quickly, so that's a careful process. That needs to be then converted to cDNA, typically because that's more stable. Then for sequencing, we need to go through library preparation, which again depends on the sequencing approach, but that also has effects on the cDNA that's being processed. We have sequencing, typically cluster generation on a MiSeq or a NextSeq in our case. Then we have quality control of the sequencing data. Typically, we may map this read or do pseudo alignment of those reads to a reference or a bunch of references. That will allow us to quantify how many reads are present per transcript. That information can then be used to quantify their relative abundance and the types of isoforms that are present. And that maybe, if it's relevant to us, allow us to compare between the activity rates of certain genes in different conditions. So linked to that, we have to think about for the wet lab component then, how are we going to isolate the typically it's mRNA that we want? How many replicates are relevant? Biological replicates are far more important for RNA-seq because we're looking at a dynamical system that changes a lot. Technical replicates are not very useful. Batch effects. If we do many small-scale experiments over different different time periods, say between different years or different months, that is something to consider because kits, protocols, and reagents may change. Historically, unstranded libraries were used, but they're not really common. And at the ProBite, we just use stranded libraries only, so that's not an issue. Read types, longer reads are better, but for RNA-seq, their quality will decay from 5' to 3'. And then we also have the sequencing depth that is possible. And especially worth noting that for RNA read libraries, QC is very, very important because RNA tends to degrade, whereas if you're dealing with DNA as your original source material, that will degrade much less. So here we're talking about bulk RNA-seq, not single-cell RNA-seq. So that means we've got a pool of cells and the associated genes at the start. Up here, we prepared the libraries and all the steps to apply the different reads. And this bundle of short reads are then aligned to the genome. Typically, they align at exons much better than they will at introns, but it depends on the tempo of mRNA that we're dealing with. There are two main approaches after that. One is alignment-free quantification, where we would use a tool called Callisto and Sleuth to examine that, and that uses a form of pseudo-alignment to quantify the level of the number counts of each at each isoform. And then a slightly different approach is to count-based methods, where we're trying to quantify based on the numbers of reads mapped to a genome. So the whole read is mapped, in contrast to alignment-free approaches that use a partial alignment method, and that's counted up. So for RNA-seq experimental design, we have to consider that we might have steps associated with mRNA enrichment and indeed PCR at the start up here. We may have either polyase selection or ribosomal RNA depletion. We'll then have library construction if we're using the nanopore approach, which maybe look more rare here. There's different kits and different approaches for that, in contrast to Illumina, MiSeq, or NextSeq, where there's certain established library preparation approaches. This will lead us to the situation where we have a sequence read library and we want to explore that further. So what we're thinking about here is these general steps that we have RNA at the start. We create a cDNA library from that, we sequence it, and our goal is then to estimate what isoforms are present. So that's our first question. And then what relative levels do they have? So it's two kind of questions that we need to think about. And for these isoforms, we have to think about it very carefully. So if we have some sort of intron exon pattern like we saw on the other the other slide, and it looks something like this, we have to consider that when we're amplifying reads, we're just making effectively doing a random sampling across these regions of different reads that are from these portions. And the reads may have, typically they're going to be paired end. So each of these lines represent a paired end read associated with these different exons, but the numbers and frequencies will vary. And we have to consider instances where we may have, if we look at host and vectors, if we have alternate splicing event and so on. So there's quite a few things to consider here. So what to remember is that this process is a sampling process. It's a pseudo-random sampling process. It is inevitably slightly biased by the protocols that we're using, and that means if we repeat this process, the distribution of reads that we get will vary at every experiment. So if you think of a plot like this, we do the same sampling process and then look at the same another experiment for the same set of reads, the same sample, the same sequencing, but the same implementation of the protocols, we'll find that it's not quite the exact same. It's slightly different, and there's some sort of different features. Hopefully not that big. My pen moved, but you'll see that slightly different every single time. And that's worth bearing in mind. RNA-seq gene expression is dynamical. It is a system that is not static. Gene expression is seldom a process where we have constant expression. It varies, and that's different from genome sequencing, where effectively we do have something that is pretty much static. So consequently, genomics and transcriptomics are quite different. So what are we trying to do when we're looking at RNA-seq data? Well, we're trying to think for each CDS they have, we find to which CDS or isoform each read belongs, and can we identify the exact region in the CDS? Additionally, we have to consider that there's a lot of data transformation manipulations that are underway. So when we consider this process, this pseudo-random process of the non-random generation of different reads at these isoforms, can we get the numbers of reads originating at each isoform? So you can then compare between the different isoforms to see to what extent are they expressed. So there's a good article reference down here with this kind of layout, which gives a good indication of the main steps in this process. Mentioned before that we have experimental design, which we're going to concentrate on here. To think about these parameters that can address our sequencing research question. We have the sequencing generation, the FASTQ file. we do QC here, which is very important. We're then able to do mapping the reads, assuming we have no contamination. Then we can move on to counting the reads, followed by quantification of the relative abundances, differential expression, and then finally the visualization and summarization at the end. So that's kind of our approach, irrespective of what kind of tool we actually use. So that's summarized again on this slide, taken from this paper that also might be useful. So we can use things like FastQC for checking the quality, FastP for retrimming and adapter removal. If we're working with some weird vector, then you might want to do de novo transcriptome assembly, where RNA spades and Trinity might be good options there. If we're looking at read alignment and quantification, we have two approaches. We can use a mapping-based approach with Minimap2, and then evaluate that using HT-seq. Or alternatively, we can use pseudo-alignment with Callisto and use Sleuth then to process that information and interpret that to perform differential expression analysis. Another option then is to use TX import, which can work between different tools. So for example, we could use limavoom instead to perform a linear model of the gene expression. So that's kind of our approach. We have to think about our first part of the experimental design. And what's really, really important for this is to think about how we can decrease uncertainty. So if we're trying to estimate our mean expression level per particular gene in a particular condition, we have to think about that if it's a single estimate from a single value here, we might have more information if we have a second estimate. So 2 biological replicates can improve our power. And likewise, as we go to 3, 4, 5, what we find is that the standard error associated with our mean expression level for that particular transcript decreases. So remember that your standard error, SE, is equal to the standard deviation by the square root of n. So as n increases, our standard error decreases. So this improves our certainty. So note here, in each case, we're talking about biological replicates. So you can ask yourself then, how many biological replicates are in this experiment? And this is taken from this website below. Equally, if we have something similar, so you've got 6 samples here again, how many biological replicants are present here, for example? So again, you can think that through, and this might be something you come across in the lab quite often. So what we can say about biological replicates is that technical replicates come from the same biological organism or indeed particle, where we're taking the same repeated samples from the same entity. In contrast, biological samples are different biological replicates. Maybe sampling independent cultures, independent mice, independent hosts, and that provides more information relevant to RNA-seq analysis. So if we're looking at technical replicates, we'll find that they're nearly all uniform. There should be very limited variation. Whereas with biological replicates, we can capture the biological variability. So if we're looking at protocol development, for example, technical replicates can be useful because that will tell us if we have issues with our protocol. We can then subsequently apply these protocols once they're developed and streamlined to biological samples, because that will tell us more about the biological question that we have at hand. So both can be useful, but for RNA-seq, if we have a biological question, we have to think about biological replicates. So to address that question, technical replicates won't help you address that. And the biological variation is much, much higher. And it's always better to, where possible, sequence more samples rather than more technical replicates. There's an illustration from this article by Liu et al. From some time ago, where we look at the numbers of reads versus the number of detected differentiated expression genes given a false discovery rate of 0.05. And what we see is that it increases irrespective of the number of reps that we have. But what you can note is that the jump, say from 2 to 4 to 6 replicates corresponds with roughly a doubling in power, or a substantial increase in power each time. Whereas the number of sequencing reads, sure, at a very low level, which we're unlikely to encounter, it does increase. But once we go up to higher levels, and it's likely that you actually get more than 30-fold average coverage in your experiments, the increase in power is small. So biological replicates have much bigger enhancement of power than for more depth. Of course, we should be aware that power testing for RNA-seq is not simple at all. And in essence, the distribution of expression values found in transcript is not the same, and each has a different variance in mean expression levels. So in theory, we should actually do the model for each isoform. And we also assume that each isoform is independent, which is not true in reality. They're partially correlated. So these are things we need to bear in mind when doing these experiments. So the message I'm trying to get across here is that the capacity to detect changes in expression is a function of the number of samples per group. So it's not the cumulative one, it's per group. And we should be aware that the group with the fewest samples is the most limiting, has the most limiting effect on power. So allotting numerous additional samples just to one group only won't actually have as much increase in power as you might imagine. So what we can use is this calculator at this website here. You can click on it, and then what you can put in is the number of experimental subjects you want to look at, the number of the average read counts, which might be 25 or 50 or 100 even. The dispersion level is the amount of variance in expression here, which you can just leave at 0.5. Same with the normalization factors, you don't need to change that. The number of genes we want to test will change, and we'll look at the interaction that might be prognostics. It's reasonable to leave this at 100 and to reflect that in the false discovery rate. And we can also require adjust the minimal fold change required from 2 to something else if we want. And we should note that that's the actual biological effect. So if we want to detect more subtle biological effects, we need more samples. That's clear. So if we look at examples here, what we see on the left is that as we have a higher sample size, if we have an experiment where we have an average of 25-fold coverage and use a threshold of at least 2-fold coverage up or positively or negatively, we can see that if we've only 15 samples, we have minimal power to detect true changes. That basically we're looking at noise. And as it increases, then we can see we pass the threshold of about 50% over here. Maybe that's 36, about 36. And then if we want 80% power, we're looking at over 50. What we can do to adjust this is increase the full coverage threshold. So you see a lot of studies, people will decrease that. That's a bad idea. That's bad practice. We want to avoid that. So here, if we look at these two options, here is a coverage of 100 and the full coverage threshold is greater than 2. So you can see the coverage, if we go back to the 50% threshold, coverage helps a bit, but it's not really going to change it dramatically. Whereas if we make the full coverage threshold more stringent, you can see now that the number of samples that we need to achieve that threshold is now much less. So it can be difficult to get samples. There's budgetary and logistical constraints on that. So an option to mitigate that is to use a more stringent full change threshold. Now, you may think that will miss genes that have changed in expression, but in reality, you don't have, the experiment doesn't have power to detect these. So, you'll just be, if you use the thresholds below that, you'll just be looking at noise. It won't actually have any biological meaning. It won't answer your question that you're trying to ask. So what we can say is that, for example, if you have low coverage and use a full chain threshold of 2, only 35 more samples per group to have 50% power. If you have higher coverage, that will drop to 28 samples. If you want, if you use a higher full chain threshold, that drops to 8 samples. That might be more realistic for many of you. And If you want more power, so you want to go up to 90% power, then the number of samples you need in addition is not too, it's achievable, perhaps in certain contexts, 12. So let's look at examples. So we can look at examples for cows and pigs and Anopheles gambiae, just as an example vector species. So what you see is that the number of cDNAs differs for each of these, and that will mean our capacity to look for its expression is slightly different. It's not the exact same for each of these. And what we're going to assume here is that our false discovery rate is 0.05 for each of them. And we assume for each of these transcripts that 5% have may have changed expression. So that will depend on your setup. And here we're assuming that we're just comparing two groups where we're considering the number of samples in each group. So you got two groups, maybe that's case, and then the other ones are control, and we want to compare them to each other. So if we do this for Bos torus, firstly, we have listed our assumptions here: 50-fold coverage redepth, 5% our differential expression, and an unbiased dispersion. If we look at a fault train threshold of 1.5, which you will see in some studies, which is a bad idea, you'll see you really need large sample size, which seldom do we actually have. A fault train threshold of 2, which should be people's default. Again, we need quite a few samples for that. If you use a more stringent fault train threshold of 3, you can see that the number of samples is a bit more achievable. So you can zoom into that sample size of less than 10 on the next slide. What you see there is that we've got 4 different thresholds. So we go from 2.5. So if we have small sample sizes and we've got a fault train threshold of 2.5, we've limited power. We're in this region. If we increase it to fault train threshold of 3, we need about 9 samples to 50%. If we increase it to 3.5, we need about 7 or 8. If we increase it to 4, we need 6. So that kind of gets in 6 per group, so 12 in total. So that kind of tells us about how much power we'll have if we use this threshold given a typical RNA-seq experiment. For Scrofa, this is the summary of the numbers needed. So if we use 80% power and we want fault train threshold of 2, we need 37 per group. 50% power, that drops to 25. A fault train threshold of 3, that drops to 13 for 80% power. For 50% power, that drops to 9. For 80% power with fault train threshold of 4, that's 9. For 50% power, that drops to 6. For Scrofa, it's similar, as you can see from this diagram here. What we see here is that our power is slightly lower. So you can see there that passes the 50% power threshold around here. So about 35. What you see when you zoom into the samples less than 10 is that for 3, we have fault train threshold of 3, we don't have much power. For a fault train threshold of about 3.5, we need at least 10 animals per group. And then for the fault chain threshold of 4, we need about 7, at least 7 animals per group. So that's summarized in this slide. You'll see if you have more isoforms that are theoretically present, we need more samples per group compared to what we have for Bos torus. So you can see there, if we go down, we want just 8 samples, we have to just be aware that that will correspond to having 80% power if we use a fault chain threshold of greater than 4, assuming 50-fold average read depth. In contrast, if we look at a vector species like Anopheles, got 50K isoforms, our power will be a bit better. So here, when we look at about 50%, we're looking at here 25-ish. If you were to look at the low sample number options, you can see that if we look at 50% power for fault train of greater than 3.5, looking at 9 samples. If we want 50% power for fault train threshold greater than 4, we're looking around 8 samples. And that's summarized here. You can see that this is less than the sheep overall. And say if we have 10 samples per group and use a fault training threshold of 4, this corresponds to 80% power. Now, you may be asking, so what if I just increase the sequence coverage? This will only have substantial effects if we have very low coverage. So here we've got the original data set with 2 replicates, and you can see its level uncertainty as a function of sequencing coverage on the x-axis. It does drop down a lot at the start, but then it tails off. And if we're going to be out here for our experiments, you can see it's not much of a difference if we go from 25-fold coverage to 50-fold coverage. If we double that in 4 replicates for the same read depth, we'll see that we get a big fall in uncertainty, much better power. Whereas if we increase the coverage for them, it's not a big jump from 25 to 50. If you have 2 replicas, but you double the read depth, we can see that power is higher, but again, it's not the magnitude of change. It's not as big as if we just double the number of biological replicates. And we only really see effects if the read coverage is very low. If it's very high, it makes no real difference. We also have to bear in mind what we're trying to do. So with the ENCODE consortium has suggestions on numbers of reads that we need per sample. So we're assuming here that we're all using reads that are longer and they're paired end and stranded. They talk about looking at, if we want to look at gene level differential expression, that we should be looking at about 15 million reads per sample. If we have numerous biological replicates, we can get away with fewer samples. If looking at isoform level of expression, we need more reads, at least 30 million per sample for known isoforms. Whereas if we're looking for novel isoforms, we need 60 million reads and perfectly long ones because we may need to do transcriptome assembly to really identify these properties. So that's quite tricky. So we can kind of contrast between new and novel isoforms and gene level expression, which is easier to detect than isoform level expression changes. Finally, what we have to also be considerate is that if we're comparing bulk RNA-seq versus single cell RNA-seq, they're quite different because single cell RNA-seq, which is on the y-axis here compared to the bulk profile, single cell RNA-seq will have its read split between numerous cells, which will mean that its distribution of values will be lower. So this is plotted on the same axis, whereas the red line is what we would expect. We'll see that some genes here have 0 expression in the bulk profile, but have some expression in the single cell profile, but relatively more have 0 values for the single cell profile and some value for the bulk profile. So this is as part of the sampling process. Genes with low level of expression will be harder to detect, and what we should be aware of is that if we do single cell sequencing and compare that to a bulk profile, they won't agree exactly. And what we expect at low expression levels is that there'll be a deficit in expression for single cell outputs. So it's just worth bearing in mind. So in summary, what we're saying is that RNA-seq experimental design is not trivial. It needs thought and planning. We know that biological replicates, where possible, improve your power. Read-up does less so. So it makes an extra effort to do fewer, larger experiments will benefit you. If you look at experiments, sometimes the biological effects are so large that using numerous samples will be not a good use of resources. For RNA-seq, that the power profile is different, power estimation is different, so it's not the same in essence. What this suggests is that we should focus on fewer, larger RNA-seq experiments where you have numerous samples so that we can answer the question, and that if we do many small experiments and try to link those together, that will create issues, and it's not wise. So fewer, larger experiments is where we want to be. If you're not sure what to do, BSP members are here to help. So Graham is very knowledgeable about RNA sequencing approaches and in different options. Has great experience in that area, as does Naomi Bolo. Chandana Tanakun also has extensive experience in dealing with RNA-seq from the bioinformatics end. I have some experience in terms of dealing with RNA-seq profiles as well. And then Nikos Angelopoulos also has experience with differential expression. And it's modeling that, integrating that, and extending that to perform more powerful analysis. So please ask us if you want further advice on this area. And please see the reading material in the document also in this folder.